Hello and welcome to lecture number 3 in our series on physiological psychology. Today we'll be talking about the synapse and synaptic potentials. We'll talk basically about how the action potential gets generated at the dendrites, and we'll talk a little bit about how excitatory and inhibitory postsynaptic potentials can increase or decrease the probability of an action potential occurring. So the concept of the synapse was first discovered by Charles Scott Sherrington in 1906 to describe the specialized gap that existed between neurons. So neurons communicate by transmitting chemicals at junctions called synapses, and synapse is just Latin for gap. So Sherrington's discovery was a major feat of scientific reasoning because he basically deduced the existence of the synapse through his data. So he wasn't able to actually observe the synapse, but he was able to deduce that it existed based on his data. So what Sherrington did is he investigated how neurons communicate with each other by studying reflexes, which are automatic muscular responses to specific stimuli. So your doctor has done this before. They tap your knee and your knee pops out. So this is in a process that occurs as the result of what's called a reflex arc. So the leg flexion reflex occurs when a sensory neuron excites a second neuron, which excites a motor neuron, which excites a muscle. So in a four-legged animal, you can poke one foot, and they will withdraw one foot and then relax the other 3 feet. So this is known as the leg flexion reflex. This occurs through this reflex arc. So we have the activation of receptors at the skin that travels up a sensory neuron to an intrinsic neuron in the spinal cord that then travels back down the motor neuron to cause the flexion of the muscle. 3 important points to know about reflexes. Reflexes are slower than conduction along a single axon. We also know that several weak stimuli present at slightly different times or slightly different locations can produce a stronger reflex than just a single stimulus. And also, as one set of muscles became excited, another set relaxed. So that tells us very important things about why reflexes told us so much about how neurons work. Because what's happening is we get excitation along one path and inhibition along another. And so as a result, we get differing responses based on that reflex arc. We also know, because they're slower than conduction along an axon, that there's something slowing down that process, and that process is this communication across the synaptic cleft. So if we have the speed of conduction along a single axon being 40 meters per second, the speed of conduction through a reflex arc is much slower and more variable, sometimes 15 meters per second or less. Presumably, that delay occurs at the synapse because it takes time for information to cross the synapse. The first concept I want to introduce in this area is what's called the excitatory postsynaptic potential. We have to get a little lingo out of the way first. We talk about a presynaptic neuron as a neuron that delivers the synaptic transmission. The postsynaptic neuron is the neuron that receives the message. So when we talk about an excitatory postsynaptic potential, we're talking about excitation occurring at the postsynaptic neuron, which is at the other side of the synapse. So it's receiving a message from the presynaptic neuron, which is delivering the message. So these excitatory postsynaptic potentials are graded potentials that can decay over time and space. So the cumulative effect of excitatory postsynaptic potentials are the basis for both temporal and spatial summation, which we're going to talk about here in a second. So remember what Sherrington found is that if you use the same level of stimulus that on its own would not produce a reflex, but did it several times in a row or at several different locations, you got a reflex, and sometimes at a greater magnitude than you might have from just a single sufficient stimulus. So what Sherrington discovered are temporal summation and spatial summation. So Sherrington observed that repeated stimuli over a short period of time produced a stronger response. Remember, an action potential will only be generated if a cell is depolarized past the point of threshold. So when we have several small depolarizations that are not sufficient to generate an action potential on their own, they will summate over time. That is, they don't diminish all at once. So while a single excitatory postsynaptic potential that is not sufficient to cross past the threshold will not produce an action potential. However, several in a row might, because the earlier EPSPs have not worn off yet, because they take time and space to depolarize. So we get this idea of temporal summation. So repeated stimuli can have a cumulative effect and can produce a nervous impulse when a single stimulus is too weak. So the idea of temporal summation is stimulation at the same location in a sort of rapid amount of time, so there's insufficient time for the EPSPs to wear off, that then allows for the summation of these EPSPs at the axon, and then that generates an action potential. Spatial summation occurs when we get stimulation at several different locations. So Sherrington also noticed that several small stimuli in a similar location produced a reflex when a single stimuli did not. So not the same location, but very nearby. So this effect is known as spatial summation. We get synaptic input from several locations, which can have a cumulative effect and trigger a nerve impulse, because they are summing on the same neuron coming from several different locations. So to give you an idea what this looks like, we might have a single EPSP, which is insufficient to cause an action potential. Then if we have several in a row, maybe two, not sufficient to reach the threshold of excitation, which you can see from that dotted line. Whereas in the third, we get 3 EPSPs that combine, and that then exceeds the threshold. You can see that an EPSP sort of is diffuse over time, and so while it hasn't worn off yet, if another EPSP arrives, it's going to push that neuron further up and further up until we cross the threshold of excitation. Whereas when we get to 4, we get simultaneous EPSPs that combine spatially all at once to exceed the threshold. And similarly, then we can get IPSPs, which we'll talk about here in a minute, which are inhibitory postsynaptic potentials. So spatial summation is actually critical to brain functioning. Each neuron receives many incoming axons that frequently produce synchronized responses. And this synchronization is an important part of how our brain works. Temporal summation and spatial summation often ordinarily occur together. And so through this temporal and spatial summation, we're able to process lots of complex information by producing various kinds of information over time and space. So you can see we might have spatial summation, where there is impulses arriving from several neurons at the same time, or temporal summation, which is several impulses from one neuron over time. To give you an example of kind of how this might work, let me draw you an analogy. So imagine that you're standing on the edge of a cliff, right on the threshold. So we're talking about the threshold of excitation. And someone pushes you just a little bit. Not enough to push you over the threshold to fall down the cliff, but a little bit. That would be kind of like an excitatory postsynaptic potential that doesn't push the amount of depolarization enough over threshold. Now imagine that same person pushes you the same amount, but 3 times before you can recover. So there's a good chance that you're going to fall over, because you will not have recovered from the first shove, and so you're still off balance, and so they push you again, and you're really off balance, and then one more time, and you actually go over the threshold. That would be an example of temporal summation, where it's sort of 3 incidents in a row cause you to cross over the threshold. Now, by analogy, sticking with the same analogy, imagine spatial summation. So rather than one person shoving you all at once, 3 different people shove you all at once, pushing you over the threshold. That's how spatial summation works. So temporal summation and spatial summation can work. So it might be 2 people, or one person shoves you a couple times and one shoves you once. You know, all of these things can combine together. And that's how temporal and summation, it's a kind of an analogy that you can think of when thinking about temporal and spatial summation. So one thing we haven't talked about is an inhibitory postsynaptic potential. Sherrington noticed that during the reflex that occurred, the leg of a dog that was pinched retracted, while the other 3 legs were extended. This suggested that an interneuron in the spinal cord sent an excitatory message to the flexor muscles of one leg, and an inhibitory message was sent to the other 3 legs. So what this indicates is what we call an inhibitory postsynaptic potential, which is the temporary hyperpolarization of a cell membrane. Now remember from our last lecture, when a cell membrane is hyperpolarized, that means it's harder to generate an action potential. So an inhibitory postsynaptic potential is the temporary hyperpolarization of the cell membrane. This occurs when synaptic input selectively opens the gate for positively charged potassium ions to leave the cell, or negatively charged chloride ions to enter the cells, thus generating an inhibitory potential by hyperpolarizing the cell membrane. So this serves as an active brake that can suppress excitation. It's also the way in which a number of drugs can influence our behavior by causing IPSPs. So for example, the inhibitory neurotransmitter GABA can allow chloride ions to enter the cell, thereby hyperpolarizing the cell, thereby making it less likely to generate an action potential. So these inhibitory synapses occur also in the spinal cord, just like the excitatory synapses. So how does this all work together? How do these have relationships amongst IPSPs, EPSPs, and action potentials? Well, Sherrington assumed that synapses produce on and off responses, and that synapses vary enormously in their duration of effects. So the effect of two synapses at the same time can be more than double the effect of either one, or less than double. So there's really a lot of variation in the way synapses work together. So one of the things that we know about IPSPs and EPSPs is they have an effect on what's called the spontaneous firing rate. So each neuron has its own spontaneous firing rate, which is the periodic production of action potentials despite synaptic input. So it's basic, they just periodically fire. Excitatory postsynaptic potentials increase the number of action potentials above the spontaneous firing rate, whereas inhibitory postsynaptic potentials decrease the number of action potentials below the spontaneous firing rate. Now Sherrington noticed this as a function of reflexes, but we'll see that Hebel and Vasel and others have discovered that in our visual system, this is actually an important part of how our visual system works. So we'll talk about what's called lateral inhibition, where nearby cells inhibit, or cells inhibit nearby cells, such that we end up inhibiting nearby cells in the visual system. This creates some important visual illusions like Mach bands, and we'll talk about what's called Hermann's grid. And that's all due to this change in firing rate because of EPSPs and IPSPs. All right, well that's it to our introduction to synapses. Next up we'll talk about this process of neurotransmission.